actually missed the deadline on the title, and it's a little bit different. So I switched methods halfway through, and it's actually investigating the population structure of a hardwood-infesting coastal bark beetle. So we're switching gears slightly, but still in the same system. So I'm going to talk about the general life cycle of bark beetles and what we know of in conifers first. So there's three main stages. There's colonization, breed production, and emergence and dispersal. So within these three stages, there's substages, of course. So within colonization, you have pioneer beetles that are orienting to trees based off of olfactory cues and the shape of the tree. Host acceptance occurring if a beetle decides that it wants to stay in that tree based off of gustatory stimulation or taste testing to determine the quality of the host tree and also the amount of defensive compounds that are present. If it decides that this is the tree that it's going to stay in, then it undergoes conspecific attraction, releasing aggregation pheromones. In brood production, you have mating occurring between males and females, maternal galleries being constructed underneath the bark, as well as larval development, where you have larvae feeding on the subcortical tissues or the phloem of the tree. In the last stage, we have pupation occurring, which finishes the holometabolous development of the bark beetles, emergence out of their natal tree, and then flying to a new host tree as that natal tree, the resources are being depleted. So for the population structure, again, of what we know of in conifers, we know that bark beetles sexually reproduce, and that's the sex ratio of sexually reproducing animal species tends to be 1-to-1 or 1 male to 1 female, which is called Fisher's principle. So this is a principle that males and females approximately cost equal amounts to produce, and so the probability of an individual persisting in a population depends on the opposite sex's prevalence. This is kind of just evolution theory. So we know that there's a couple different mating strategies in bark beetles. There's monogamous bark beetles, where you have 1 male to 1 female, and polygamous or polygyny happening, where you have multiple individuals. So in bark beetles, commonly it's seen 1 male creating a nuptial chamber and then females being recruited to that area. So what about if you don't have this nice 1-to-1 population structure? So this is when you would have a skewed population structure. So a couple examples of this, of why you wouldn't have 1 male to 1 female. The first one being sexual size dimorphism. So this is when you have the different sexes being different sizes. So this can lead to very temperature-induced mortality. So there's a study done on Dendroctonus ponderosae, or mountain pine beetles, where the females were slightly larger than the males. And so as the cold temperatures persisted throughout the winter, you just had more males dying as they were smaller, they had fewer energy reserves, and so then the sex ratios started to become more female biased. Another example is that of spatial constraints. So if you think about the amount of volume or space that's present between the bark and the sapwood, if you want to maximize your fecundity or the amount of offspring that you can have, then you can slightly modify the population structure. So this is called local mate competition, so it's only producing a limited number of males to reproduce with females, which is sibmating. A more extreme case of this is haplodiploidy. So this is the determination of an offspring's sex based on the fertilization choices of the mother. So female offspring will be created from fertilized eggs, meaning that they are diploid individuals, and on the contrary, males are created from unfertilized eggs, meaning that they are haploid. So this is an extreme case, and it's seen in Xylosandrus germanus, which is an ambrosia beetle species. So I've been talking about conifers. What about hardwood infesting bark beetles? So this is very vastly understudied. We don't really know a whole lot about hardwood trees and their consequent bark beetles. Trees are also very ecologically important in the hardwoods. They are early succession trees. They provide lots of nutrient inputs to riparian areas, and also lots of diversity. So this talk will be looking at the alder bark beetle, which is Alnophagus aspericolis, which is a native primary bark beetle that infests the nitrogen-fixing red alder host trees. It also is kind of an interesting bark beetle because it has a potential mutualism with a non-ophiostomatoid canker-causing fungus. So it makes it kind of a unique one to look at. So this talk will be using the alder bark beetle as a study subject to better understand the life histories in hardwood infesting bark beetles, so kind of switching gears from the previous talks. So this is a proposed life cycle that was created based off of previous research by John Borden in 1969 and Wortman et al. Recently in 2022. So we found that there are beetles present overwintering in the branch nodes, and these are my scribbles as well. In the spring, when temperatures warm up, they disperse to the bowl. They then undergo colonization and brood production, and then spend most of the summer feeding, trying to get big and strong as larvae, and finishing it off and moving up to the branches and that second generation. Additionally, we're seeing that in another group, you have beetles that are present as larvae overwintering in the bole or the stem of the tree. So they're present as a late instar or a late stage larvae. When temperatures warm up in the spring, you have them finishing their larval development, dispersing to a new bowl, kind of just seeing that it's offset from that first group, undergoing colonization, brood production, and then larval development as they're shifted. So you can see that there's two different groups, the A and B. However, there isn't any speciation occurring. There's still one species of alder bark beetles, so there must be some sort of gene flow. So you can also have this case here, where potentially you have a colder summer happening in a poorer resource tree, and so then these beetles, rather than finishing their whole development and moving up to the branches, would be finishing in the bowl. Same thing, opposite, I guess, happening, where you have beetles from that second group finishing their development and then moving up to the branches. Also note here that we have two different overwintering states. We have beetles that are present in branches and then also present in the bowl. So this is what we wanted to look at for this research. So for the methods, we wanted to look at the population structure between the branch overwintering beetles and the bowl overwintering beetles. So we went out and collected a whole bunch of branches that had evidence of alder bark beetles, so frass and entrance holes. We pulled the branches apart, pulled the beetles out, and then sexed them. The bowl overwintering beetles are a little bit trickier because they are larvae in the early spring. So we went and cut into trees and found ones that had larvae in them. We put emergence traps, and then we sat and twiddled our thumbs and waited for the beetles to come out later on in the season. So you might be wondering, how do you tell male versus female alderbark beetles apart? So it's not as easy as the Dendroctonus species that have nice stridulation organs that make noises when you bug them. These ones we actually had to remove the elytra and look at the abdominal tergates. So females have 7 tergates, where 8 tergates are seen in the males. So what did we see? We saw that for the observed sex ratios, the bowl had 71% of the population was female, taken from a sample size of 442 beetles across 9 traps, 3 sites, and about 5 months of observations. In the branches, we saw that there was 80% females with a sample size a little bit smaller, but 106 beetles of 20 plus branches and 1 site. So for the stats, we found that the male versus female proportions were statistically different from the expected 1-to-1, as Fisher's principle was stated before, with a statistical p-value. So that's comparing if we were to have a 50% male versus female ratio. We also found that the frequency of females between the 2 overwintering sites, so these 2 numbers, were statistically similar with a p-value greater than alpha of 0.06, and both of these were done using a chi-squared test. So in conclusion, the sex ratio of alder bark beetles is heavily female biased, as those past graphs very obviously showed, in both of those overwintering locations, which is pretty interesting. Not really uncommon for bark beetles, but interesting to see. As the proportion of males to females doesn't differ between the different overwintering locations, the sex ratio bias is most likely not a result of differential activity, or so for example, dispersal propensity, where propensity means an inclination or a natural tendency to do something. So initially when we looked at the system, we thought, oh, there's more beetles, females in the branches, perhaps they're flying up, but we found that the same amount of females or percentage was also found in the bowl. So why could this be happening? What are some reasons? I mentioned a couple of them earlier, but we could have polygyny happening, where you have individual males mating with more than 1 female, so potentially skewing that population to be more female biased. You could have local mate competition occurring, so that's SID mating, or the extreme case that's seen more commonly in ambrosia beetles of the haplodiploidy. Here's one that I haven't talked about before, but you could have endosymbiotic reproductive manipulators. So these are parasites that are present in different arthropods, and they're passed on maternally from female to female. And so they do something really weird where they can actually skew the sex ratio by causing higher rates of mortality in males. They can impact the cytoplasm of the sperm, deeming males sterile, and doing a whole bunch of wacky things to increase their host abundance in females. Lastly, one that I mentioned already earlier, you can have sexual size dimorphism, where you have size differences between the males and the females, meaning that you just have more females or males, depending on what's that. But I don't think this one is the case, because they're pretty similar in size. Okay, just to finish it off, I'd like to thank my funding sources, NSERC for giving me a CGSM for funding this year, as well as the Faculty of Forestry, my regional parks, and the research forest at UBC for letting me put traps up, take beetles out of their forests, and then study them, and of course my research lab. So thank you.